Thank you, Jackie, very much. This is Barry Bradford from Michigan State University, and really pleased to invite our next speaker up to the podium, virtually, if you will, and want to introduce Dr. Teresa Casey. So Dr. Casey earned her BS in biology from the University of Connecticut and completed her master's, PhD, and postdoc work at the University of Vermont. Right now, she's a research associate professor at Purdue University, and she has a research program focused on understanding the regulation of mammary development and lactation from molecular up to systems levels. Currently, she's aiming to understand a role that circadian clocks play in the regulation of mammary development and lactation, as well as in maternal environment effects on lactation and on neonatal growth and development. So we're really looking forward to Dr. Casey's presentation, and please take it away. Oh, you're muted.
Thank you for having me, Dr. Bradford and Bowerman, talk today. And I want to start my timer so I'm not late. And so I just wanted to give a couple of slides on saying why we started in this area of research. As a mammary gland lactation lab, we're interested in what are the physiological adaptations needed to support lactation and how are these physiological adaptations regulated in the dam. And it's the second question here that brought us to circadian clocks. So we know that hormones play a central role in mediating a lot of the metabolic adaptations in the dam across the multiple tissues, but we had hypothesized that other systems were involved in this, but we didn't know what those were. So we decided to take an untargeted approach, and this was about 12 or 13 years ago. And we hypothesized if that gene that changed commonly between multiple tissues between late pregnancy and the onset of lactation, but if we queried those genes, we may find systems that may be coordinated the metabolic changes across the dam. And for these studies, we used rats and we looked at them in late pregnancy and at the onset of lactation. And this is just a beautiful picture of what happens in the mammary gland as it transitions between pregnancy and lactation. And in addition to the mammary gland, we looked at adipose tissue and liver tissue, and we asked what genes may be commonly changed between them. And what we found is that among the three tissues, that these core clock genes were changing from pregnancy to lactation. And this is the fold change between them. And in general, we saw a set that were increased commonly and decreased commonly in the three tissues. So then we asked ourselves, could circadian clock genes be homeoretic regulators? Do they regulate processes that ensure and maintain homeostasis? We do know that clocks play a central role in regulating homeostasis, so they fit that role. And are they able to respond to physiological and environmental challenges and affect growth rate, tissue differentiation, and metabolism? And at that time, or around that time, there was really some beautiful work coming out of the University of Aberdeen in Scotland, in which they showed that the clocks definitely regulated different metabolic levels according to the photoperiod. And I just encourage you, if you're interested in it, it's beautiful, beautiful work that they showed how the clocks change metabolic rate across multiple tissues to modify growth rate by length of day. So it fit categories that it could be. So we did a little bit of work to look into it. And what is the circadian timing system? Well, it's a system in our body that times us, that gives us time. It does that by generating 24-hour cycles of biochemical, physiological, and behavioral processes. It evolved to coordinate our internal physiology. So it generates circadian rhythms of nearly everything, hormonal levels, core body temperature, everything. They originally evolved in single-celled organisms, and that was because single-cell organisms that lacked organelles needed to separate things that couldn't happen at the same time. So like you didn't want high metabolic processes and generation of free radicals to happen at the same time as DNA synthesis, because we would end up with lots of DNA mutations. This temporal separation or gating of the time of entry into the cell cycle is something that occurs in eukaryotic organisms, and our data in ChIP-seq analysis indicates that that is true in the mammary gland as well. The other reason they evolved was to synchronize our physiology and behavior to the environment. And this is really important probably to the dairy and nutrition field, is to understand how the environment and behavior affect our physiology, or when things occur during the day. I don't think I need to tell anyone that circadian rhythms are important, because three gentlemen who discovered the circadian clock mechanisms were awarded the Nobel Prize in 2017. But what we need to keep in mind is, when we think of cows, is that the majority of the work that was done, it was done in male organisms. And that we have to, when females are very different, and they're particularly different when we consider reproductive states, and about a third of the genes in our genome are regulated differently from female animals to male animals. So there's a lot of work that needs to be done when it comes to understanding circadian regulation in a female system. Additionally, most of the work were done with monogastric animals. And I probably don't need to tell this audience that cows have a very different ingestive behavior and sleep-wake cycle. There's some really great research coming out of Germany right now. I can't remember the researcher's name, that looks at the sleep-wake cycle. It's a really primary input to the circadian timing system, as well as ingestive behaviors. And so we need to do investigations on circadian rhythms in these animals, because they behave very different than monogastrics. So the circadian timing system is a system of clocks. Every cell in our body has a clock. Those of you who do cell culture research, keep that in mind. I didn't do that in graduate school, probably paying for the noise in my data. And every organ has a clock. In mammals, there is a master clock that controls all other clocks. And that master clock is in our hypothalamus. It has a primary input of the day-night cycle. And this day-night cycle comes in and tells that clock what time it is. In addition to the day-night cycle being a primary input, social activity, exercise, stress, and eating behavior all go in, and that clock decides what time our body is on. It then transmits that information to all the other clocks in our body through nervous stimulation, as well as hormones. And our core body temperature, the circadian rhythm of that, is a key synchronizer of all the peripheral clocks, because that's a primary output. So in general, keeping this in mind, we have our clocks have three main components. They have an input, the molecular clock itself, which I'll describe in the next slide, and outputs. So the master clock, the primary input, the night cycle, integrates this temporal information and sends out circadian rhythms of hormones that give us circadian rhythms of metabolism and other physiological processes. So the molecular clock, like anything that is a homeostatic system, exists with positive elements and negative elements that feed back onto it to stop what's happening, because it's reached where it wants to be. Well, the positive elements function as a transcription factor. This is BMAL. It heterodimerizes with another protein called clock. And as a transcription factor, they bind to the promoter region of genes and drive core clock gene expression. And the genes that it drives expression of include its own negative regulators, the period and cryptochrome genes, which are translated, they heterodimerize, and they inhibit the binding of BMAL and clock to the EBOX sequence, and essentially cut off their own transcription. This periodicity of positive and negative occurs every 24 hours, so that we generate these circadian rhythms. About between 5 and 10% of the genes that are expressed in a cell show a circadian rhythm and that are driven by BMAL and clock. Among them is this one, which you would be interested in, metabolism, is PPAR alpha. And this is what is believed to be the integrator and the reciprocal regulator between the circadian timing system and metabolism, particularly in the liver. We've done this ChIP-seq analysis. I'm writing the revision right now, but shows that PPAR alpha is an output in the mammary clock. So that's pretty cool for anybody who may be interested in it. Not only is it an output, but it directly regulates BMAL levels. So it's really highly integrated when we think about that with the clock. Central questions in our lab are, do clocks regulate lactation and how? And over the last several years, we've been conducting research in our lab that looked at determining whether the circadian timing system plays a role in maintaining homeostasis in cows and regulating mammary development during the transition period. So like any scientist, we decide to take a reductionist approach and knock out the circadian timing system function and see what happens in the animal. Inputs to clock, when they're applied at an inappropriate time, disrupt the circadian timing system, and they're referred to as chronodisruptors. So since light-dark cycle is a primary input to the central clock, what we decided was to try to knock out the function of the clock by exposing cows to chronic jet lag. And what we did is we had a control group that were exposed to 16 hours of light and 8 hours of dark, where a phase shifted group was also exposed to 16 hours of light versus 8 hours of dark, but every 3 days we shifted forward the light-dark cycle. This it was shown in humans and mice prior to our start that it takes 5 days for us to recover to about a 6-hour light shift when we fly transmeridially, or we impose it by turning on and off the lights in rodents. And so we decided that we would do this in cows, and we did it every 3 days to prevent the resynchronization every 5 days. So for these studies, we had hypothesized that, of course, I don't know, that if the circadian clocks play a central role in regulation of metabolic homeostasis, we thought if we disrupted them, we would cause metabolic imbalance and decrease milk production. We conducted 2 studies in successive years, and the first one asked, what is the effect of circadian timing system disruption in the prepartum period? And we started here because as a mammary gland biologist, so we did 3 weeks after being dry, but 5 weeks prepartum, because that's when the vast majority of mammogenesis occurs. I wanted to ignore involution to uncomplicate the science, I think, a little bit, and just look at this period. And so we looked at disruption and ketotic indexes. So we looked at glucose levels, non-esterified fatty acids, beta hydroxybutyrate, as well as measured metabolic and reproductive hormones and milk production to 60 days. And in our second study, we wanted to ask questions on what is the effect of circadian disruption on regulation of glucose homeostasis. And so for here, we tested this using a glucose tolerance test. And then we also looked at mammary development by doing biopsies and milk production through 60 days. The 2 studies were conducted by, the first one was headed up by my postdoc, Dr. Aridani Suarez Trujillo, and the second year, Dr. Trujillo mentored and helped Connor McCabe, who was a graduate student in Jackie Bowerman's lab, head up the second study. They were basically identically designed with 16 cows in each treatment the first year and fewer cows in the second, because we were doing IV GTTs, or intravenous glucose tolerance tests, and biopsies of mammary, liver, and adipose tissue and muscle. But I'm only going to talk about mammary here. We also did consistently sampled biweekly and sometimes every day in this study at 6 a.m. to capture glucose and BHB levels in both studies. For the first study in both studies, we the cows entered into a tie-stall barn at 35 days before expected calving. It was the same barn that was partitioned by two layers of fire retardant tarps to prevent light from being on either side. And the control side had the same light cycle day in and day out, where the phase shifted were exposed from 35 days before expected calving, or 5 weeks, to about 3 days before calving when they were moved to the maternity barn. So they were exposed to about 10 shifts here. And then after parturition, both were on control lighting. For these studies, we wanted to capture the circadian, by circadian sampling, blood levels, and so to circadian rhythms, I should say. So to do that, we sampled the cows at 3 weeks before expected calving, which was after 2 weeks of exposure, or 4 weeks exposure, 1 week before expected calving, every 4 hours over a 48-hour period to measure multiple hormones as well as metabolites, but I'll only show a few hormones today. And we also measured body temperature using intravaginally using the iButtons data loggers, and we programmed them to capture body temperature every 30 minutes. And body temperature was used to determine the function of the central clock. In the second study we did, it was the exact study design. So the phase shift had started 5 weeks before expected calving, ended around parturition. But for this one, we did all those biopsies, those poor cows, 4 different tissues, but I'm only going to tell mammary again. And we did that at 3 weeks before expected calving and 3 weeks after. And we also performed the glucose tolerance test at 2 weeks before expected calving and then 2 weeks after calving. So this is brand new stuff. Dr. Suarez is on, and we are trying to write this up. But actually, the first thing I should say is because we use temperature as a marker of function of the central clock, what I want to show you, and basically just believe me that these dots are well informed going around this curve. So we have a really nice circadian rhythm here where you can see a lot more spreading here. So the fit of the data to a cosine curve was really high here, but lost in the phase shifted animals, indicating we were successful in disrupting their circadian clocks. And you probably notice here that we have a really nice high amplitude where we lose this amplitude here. So this shows we disrupted the clocks. So right now, I'm not going to talk about phase shifted anymore, because what was really neat to Aradani is his name, or Ari, I call him, or who everybody calls him, we noticed a couple of things of difference. We noticed that this organization got way better at 1 week before expected calving from 3 weeks before expected calving. And one thing you notice here is we went to this mathematician and said, hey, we want to describe this. So they helped us, and we did second harmonic analysis. And what you can see here is kind of a saddling. And what that is the time we fed the cow. So an hour before you feed animals, you actually have a food anticipatory response of a rising of the body temperature. So we see this at 3 weeks, but we lose it at 1 week. So we say to ourselves, is this a cue from the central clock in these control cows to organize themselves, to get stronger rhythms? So then we look at the hormones, and in general, so this is cortisol, and this is at 3 weeks and 1 week prepartum, and this is serotonin at 3 weeks and 1 week prepartum. And granted, we're still writing this up, so I don't know what the reviewers are going to say. But typical of cortisol, it's really hard to capture a circadian rhythm. For a while, they thought cows didn't show them because of their sleep-wake cycle and their rumination requiring vigilance. Although Lefcourt found it using polynomial and sampling every 15 minutes. So we weren't surprised we didn't see something here. But what we were surprised at, if you see 1 week prepartum, the data really starts to fit the circadian rhythm. And you see the exact same pattern for serotonin. So we went back and we said, well, what's happening in phase shift cows? And the one thing we looked at was calving date, right? So our hypothesis is that it was synchronizing our cow for calving. And interestingly, in the first study, there was a tendency for phase shift cows to have a longer parturition. And in our control, and in the second study, there was no difference. But when you combine both, we had a difference of 0.05. So what we think is that that what's happening is with the loss of the circadian rhythmicity or the function of the clock, it can't time the initiation of parturition. And they show in monogastric animals that that does occur. And with sheep also that had phase shifting, had extended gestation periods. But the bummer of our studies, to transition, our main output was to ask, was there effect of circadian disruption on milk production? Well, we got two different responses. In the first one, the phase shift cows produced more. In the second one, the phase shift cows produced less. That was not the only difference. In the first study, we did affect glucose metabolism, but the phase shift cows in the first one had relative hypoglycemia. I don't know if you would define it by hypoglycemia, but they had significantly lower glucose levels in these transition period samples, whereas the phase shifted cows had insulin insensitivity. So they had this response here was what we would see in monogastric animals. The hypoglycemic effect is something you do see with circadian disruption. If you knock out the BMAL gene in mice, you get hypoglycemia. And you also, but that's diet effect. If you feed those mice high-fat diet, they actually end up being insulin resistant. So we're not quite sure where it is. We know you mess up glucose metabolism, but we don't know what it means. In the second study, I do want to point out that the poor milk production or the lower milk production of PS animals was related to poor mammary development in the phase shifted cows, because when we looked at their development, we looked at percent proliferation of the epithelium. And so this was after 2 weeks of disruption. It was significantly lower in the PS cows compared to the control. And then coincident to that, or what you would expect, is a lower lumen to epithelial area, and both were significant. So we had less mammary development. So why do I believe the second study and not the first? Well, I believe the first. I like the second answer better, is because concurrent to this, we were doing studies in humans and we got the same answer. So that humans, you disrupt circadian clocks with sleep disruption. So we measured sleep disruption. This was associated with development of hyperglycemia, which lots of people have shown this now. But we were the first to show that it was associated with delayed onset of lactogenesis and poor abilities to nurse the baby in the first week postpartum. So this is our second study, and this were consistent. Our work in mice you disrupt the whole circadian system with this clock delta 19. These pups, they have a normal pregnancy, normal delivery, but their pups die in the postpartum. And if you look at mammary development during late pregnancy, the wild type has beautiful mammary development at pregnancy day 16, where the mutant mice have really poor mammary development. So this fits that. So we're left with, what's the response? Hypoglycemia or insulin resistance. So if we just talk about insulin resistance, we're wondering whether the poor mammary development and milk production is associated with circadian clock disruption is secondary to insulin resistance. And Wendy Kohik has a wonderful review on this. I encourage you to look at this. Or is it primary to alterations in mammary clocks, or is it a combination of both? And then quickly, because I am running out of time, but as dairy nutrition people, you're probably wondering, how can we manipulate these systems? What are the inputs to the mammary clocks? Work that I did with Marina Antok, who really taught me everything about circadian clocks, or some. I still have lots to learn. We found with her that prolactin is a potent input of the mammary clock. We already all know that cortisol actually acts on every cell of the body. Work I did with Avi Shimei and Samir Mabijish in Israel, and the BARD grant showed that like the liver, which this was shown in sheep, that photoperiod can affect the mammary clock. And work that Kevin Harvatine is doing is absolutely beautiful work. And he's trying to dissect out about feeding time. So we know, other than the central clock, that feed time and feed input, and this would fit with those PPARs, are a really important component of regulating mammary clocks. When our first paper was this, and I feel like I hate putting something out and then you feel like you have to retract it. It does fit. It's kind of cool if you think about the circadian clock system. And of course, I couldn't do anything without the real people who do all the work in our lab. They had so much fun. Ari is an amazing, amazing teacher. He did such a nice job overseeing the project and our funding for these studies. And if you have any questions, thank you.
Thank you so much, Dr. Casey. Fascinating talk. So we've got a couple minutes. If you have a question that's popped in your mind, please type that in the chat. But to start with, this time of year, Dr. Casey, you know, obviously day length is shortening. We have some seasonal shifts. Are those shifts slow enough that they're unlikely to influence their circadian rhythms and disrupt these things? Or what's your thoughts on that?
So not my expertise, but I will tell you that Kevin did a really nice paper looking at those seasonal changes. And so I think like what he showed is that the farther away we are from the equator, we meaning cows, probably the more dynamic they are. And he showed that the biggest changes in them, and trees are exactly the same. It's the rapidly changing length of the day length is the biggest signal to changes in our metabolism or whatever needs to have. And so Kevin showed just really nice, I can't think about it, I read it, but it's really nice data. So we do perceive that. Certainly like growth of hair, all those seasonal things, right? They're likely mediated at the level of the clocks. And you know, whether it's it's interpreted here and then sent out to signals like change your metabolism, you know, get ready to hibernate is yeah, but we need to figure out how to harness it and do that. So yeah, that's a great paper, and they just model those changes.
Okay. Help me think through in the, I think it was the last cow study you did, you saw some pretty stark, I was pretty impressed with the changes in relative proliferation markers, I guess 12 versus 5% across your two treatments, but no obvious change in milk yield subsequent to that. Help me think through that. What do you think that means? I
think I said that really quickly. There was the phase shifted cows actually produced significantly less milk in the second study. In the first study, I have no idea what happened to the mammary gland. In the first study, the phase shifted animals produced more milk and they were hypoglycemic. So there is the potential that the difference in mammary development is actually downstream of the alterations in glucose metabolism. And that's why I was saying, so Wendy Kohik has a really great review that talks about, we all think that insulin doesn't do anything, although you probably know it does, because it causes, you know, protein synthesis and it's needed for all of that. But because, you know, it's not needed for glucose uptake by the mammary gland, we think, oh, it's not that important. But it's very important for mammary development. And so it might be downstream to that in the second study with lower milk production. Yeah, we had opposite responses, which was frustrating, but data's data, right?
Never happened to me. Yeah. Well, that was great. I may have to go back and watch that at 0.7 speed or something, but that's
no, I'm sorry, I talked too fast.
No, it was great. Thank you so much.